greetings. I can see that there are very new participants also, those who have joined. So today topic of discussion is soil microflora. As we all know that microflora is very rich source of microbial biodiversity, both in terms of bacteria as well as fungal diversity. Uh, so in addition to so among the soil microflora, the most commonly isolated and utilized organism is actinomycetes. Those produces antibiotics. And in addition to that, there are various other byproducts for which people explore different soil diversity, especially the soils that are contaminated by some kind of pollutants. Pollutant degrading organisms are mostly searched in that kind of ecological niche. So, with this word, I would like to initiate this roundtable discussion, and I would like other participants to join in and uh, tell us more about what they have worked on or how means what are the outputs they get while working with soil microflora. So, I would like to begin with Mr. Vikrant Negi. Vikrant ji, if you are online, please enlighten us with your research background and then if you have conducted any work on soil microflora. Vikrantji.
Uh, Good evening, everyone. Actually, I am from medical background, so we haven't done anything in the soil microbiology. We are working in zoonotic viral and bacterial infections in our lab. So we have molecular facilities and immunological facility in my lab.
Okay,
We are going into sequencing, but that work is not started yet. But we have got the facility, we have got the lab. We'll start it soon.
okay. Great to know that. So, uh, since you mentioned about the hospital thing, so
Yeah.
recently I started work on bacteriophage. Okay,
Okay, sir.
viruses that infect bacteria. Okay, so
Yeah.
they are host specific, right?
Right.
So we are trying to find out phages or viruses that can kill bacterial pathogen like Pseudomonas aeruginosa or Klebsiella pneumoniae.
Right,
So
sir.
we are trying to find out dump sites or soil sites that are where means hospital waste is being dumped or is being put quite often so that we can get such kind of microbes in that soil sample. And since
Yeah.
there are microbes, viruses may also be there. So we are trying to find out such things. Glad to know that
Great.
you are working hospital related works.
Yes, sir. Yes, sir. Even there are many hospital wastewater system in our hospital, no? So we get lot of resistant organisms in those wastewater. Though we have treatment plant, but sometime that from there we can get highly resistant strains and where we can look into, because these are the strains which are taking resistant genes from hospital to the environment.
Yes, yes, very
Yeah.
correct. And rather, I would like, where are you located, sir?
I'm in Almora. It's in Uttarakhand.
Okay. Uh, that is quite far. Anyways, so I was also looking for such means hospital drainage or wherever this hospital waste is being dumped, na,
Yeah.
there we may get such phases portion. And then if we get some specific or very effective viruses
Mhm.
that we can use against HDR, at least their enzymes we can use to degrade the pathogenic microbial membranes, their peptidoglycan layer.
Right, sir.
Yes,
We can
bacteriophage
working
based.
on that.
Yes. It is quite a good field. I think recently people have started publishing papers on degrading microbial biofilms using
Hmm.
bacteriophage.
Okay.
So biofilm forming pathogens are very deadly. You know that they escape antibiotic treatment.
Yeah.
First,
Right.
by genetic makeup themselves, they are MDR or XDRs, and then biofilm adds the another level to it to their defense system. So we cannot penetrate that biofilm, and then we can't kill those kind of bacteria.
Right. So one of work which I have done is on biofilm forming bacteria which causes ventilator associated pneumonia. So what happens in our ICU setups, no? Many patients goes on intubation of endotracheal tube. So
Hmm.
when those patients who are intubated with these endotracheal tube for more than 48 hours, there is a strong chances of these biofilm formation in these and inside the lumen of these one endotracheal tubes. So in one of our research project, what we have done, we have taken these endotracheal tubes after extubation, and we have cut the portion. We have taken the sample, biofilm sample from which was forming inside that endotracheal tube, and then we started looking for any bacterial presence there. So we got lot of biofilm forming bacteria, especially Acinetobacter, Pseudomonas,
Oh,
and E. coli. These, yeah. So these were the major pathogens which we got from the patients who were having ventilator associated pneumonia and were intubated by these endotracheal tubes. So we have worked for various, antibiofilm activities of different antibiotics, lock solutions, and chemicals. So here, when you are saying that about bacteriophage, so this can be another door where we can look into if if cocktail of these biofilm, these bacteriophage can be used against these common pathogens of biofilm forming biofilm forming pathogens within these endotracheal tubes. So there is another door where we can open it.
yes, and being a bacteriophage, no, uh, being a bacteriophage, it's
Yeah.
host specific, so it is not going to infect or create any issue to humans.
Hmm. Yeah.
So that I think is a good part. Although means I have not gone much into it, but so far I know all the phages are very host specific. So only
So,
E. coli phages will infect E. coli only, not even Acinetobacter or Pseudomonas.
uh
So we have to use a cocktail of phages
yeah,
specific to different hosts.
different one. So yeah, we are writing we are writing the this one research article of this work. Once it is ready, I'll share with you, sir, and then we'll see if in future we can
Sure,
work
sure.
on this part.
Yes. Recently, I have isolated one uh, bacteriophage from sewage water mud sample.
Hmm.
So the sewage treatment plant, na, there is a
Hmm.
secondary treatment plant there. That is the sludge that is kept for a long time. So
Hmm.
we isolated two phages.
Hmm.
Both are specific to E. coli only, and one for
Hmm.
Acinetobacter baumannii. Now we are characterizing it for through TIM, and then we'll go for DNA isolation and other characterization. So they have very unique clearance zone. This means they are
Hmm.
killing the bacteria very strongly.
Great, great, great,
So
great.
yes. So we are continuing that work. Thank you so much, Vikram sir. We'll be in
Thank you.
touch. And
Sure, sure,
yes,
yes. Yeah.
yes, and I'm mostly available in this roundtable discussion, and as well as you can get my number through Prathamesh, or I will drop down in the chat box.
Okay, sir. Okay, sir. Thank you. Sir.
Yes, glad to know.
Thank you.
Next, I would like to call upon Bharat ji. Bharat ji, are you available?
Yes, sir.
Yes, sir. So
Sir,
please enlighten us with your words, sir.
yes, sir. Actually, basically, I'm working in the area of bioenergy. So, whenever here we are, today's topic is basically we are discussing about soil microbes. So again, here especially exoelectrogens, if we can take, so it is very predominantly, especially martial areas, it is very predominant. So exoelectrogens up to 2012, I think. So nobody has that much idea. But whenever scientists have discovered to exoelectrogens, especially geobacters, so by using geobacter, so people are trying to extract the electrons, separate the electrons, or they are transferring to the anode to cathode. All these things, basically MFC work has started like that, sir.
Yes, sir. That is a good aspect actually. Whenever we think about soil microflora, we only think about some of the bacteria that are either having PGPR activity or maybe producing antibiotics like actinomycetes. So very traditional work. While the recent means, as you said, since 2012, I think Professor Bruce Logan is
Yes,
the point
yes, yes.
here. And since then, he has developed so many technology and so many other researchers have also contributed significantly. And now these exoelectrogens, means finding new exoelectrogens is quite booming. Means that research area is quite booming. So, very glad to know, sir. Can you tell us more about this, the presence of exoelectrogens in like swamp water or maybe uh, this estuary area or any
Uh,
kind of
Yeah, sir,
where they are
basically, yeah,
abundant?
basically whatever I have done, my postdoctoral work I have done at Israel. This is basically MFCs on this. So in this case, so basically we extracted, we separated, we isolated basically these geobacter from different sources, and after that we identified these exoelectrogens. After that, based on their electrogenic activity, so we tried to produce electricity at the, and second objective is again to produce the hydrogen. So basically, we have focused at that time isolation of these exoelectrogens. After the identification, after that, what is the efficiency of the production of the electrons, and after the efficiency of degradation of wastewater, simultaneously production of electricity and hydrogen. So this is the basically I have done in this area.
Great, sir. So have you worked with Dr. Soumya Pandit
No, not
in
Soumya
Israel?
Pandit is I know sir very well. I am came to
Yes, I think.
Sagarpati also once. I think
Yes,
Prasanna Kumar. I think sir, along with you, I published one book chapter also. I think you and Soumya Pandit has a number of books you edited, as per my knowledge.
yes. We are also editing. Yes.
Yes, yes, I
Great, great.
know very well.
So
But
for the benefit,
I did not work with him. I am from other different university.
okay, no problem. Just I
Yeah.
wanted to know because the name
Okay.
looked familiar.
Yeah.
Great,
Please
great.
say to
So
Soumya
for
Pandit
the
sir. Hi.
yes.
Please convey my message to sir.
Sure, ji. So for the
Okay.
benefit of our listener, two question I have for you. One is when you are saying that you are going to isolate exoelectrogens. So what are the criteria? How do you isolate? Means there is a specific media to isolate that. Because usually we start with a mixed culture, no? So how you define that? Okay, the exoelectrogens are now in pure form. And then the second one is, what is the criteria of selection? Means to some threshold level, beyond some threshold level, if they produce an amount of electricity, then you will consider them as a good strain to carry out further research. So these two questions I have. One is the isolation process, and another one is level of electricity production. Balaji,
Sir,
up to
yes,
you.
sir. At this moment, I am unable to discuss. So sorry for that one.
Okay, at least for the isolation part, sir, if you can enlighten us about how to isolate the exoelectrogens.
Sir, most of the exoelectrogens, we know it is an endobic case. So that is one of the most crucial thing we need to be considered. So after that, we need to basically check the this how much current they are producing during this process, and there is a specific media again. So again, I need to be recheck exactly which specific media I used at that time. So I didn't remember it actually now. So that's why I'm able to explain.
No problem, sir. We are
Okay,
just discussing. Yeah. Yeah. Thank you so much.
thank you, sir.
Yeah. Next, I would like Rasika. Are you online? Rasika ji? Okay. Yashika Sharma. Are you online? Would you like to say something on soil microflora? Yashika Sharma ji. I think maybe they are not available. So next, I would like to call upon Harsh Kumar Saini ji. If you are available, please unmute yourself and let us know your work or whatever you have done or you know about soil microflora. Harsh Kumar Saini ji. Harsh Kumar Saini ji, are you there? Okay, Swapnil sir. I think last meeting also you joined. Here, Swapnil Singh. Are you available?
सर मैं सर नहीं हूँ मैं स्टूडेंट हूँ और आई एम जस्ट न्यू
Okay,
इन दिस so I'm joined for getting idea about soil microfauna.
okay, okay. So, on which topic you are working on, Swapnil?
सर अभी तो मैं जस्ट बीएससी के लेवल पे हूँ मैंने कुछ पढ़ा भी नहीं है मतलब आई एम जस्ट
Okay,
सेकंड ईयर स्टूडेंट
okay. चलो तुम्हारे बेनिफिट के लिए बता देते हैं फिर कुछ चीजें जो मतलब you may try to look into if you are
हाँ मतलब
interested
मुझे
in.
थोड़े फील्ड का मतलब अभी जितना पढ़ा है उतना तो है बट ज्यादा क्या हो सकता है तो इसलिए ही मैंने ये आज मीटिंग ज्वाइन की
हाँ हाँ हाँ, correct, correct. No problem. So, for students like you, basic information, I should give a brief on that. So, basically, soil contains a lot of microbes. When we say microbes, microbes means microscopic organism that may be bacteria, that may be fungus, that may be viruses. Okay, all of them can be there, can be present in soil. Now, their diversity depends on uh, which area they are, means from which area you are choosing the soil. Suppose moisture content is more, then you may get more amount of bacteria. Number of bacteria will be more. If moisture content is less, you may get mycorrhiza or their spores or spore forming bacteria. Because moisture is unavailable, so they will not be growing much. Spore formers will be more abundant in such arid soil, right? And the smell that comes during the rain, the first rain that lands to any arid environment, that kind of smell is due to a compound which is secreted by actinomycetes. They are filamentous bacteria that secrete the unique smell that we get when the first raindrop goes to the soil. Right. So these are the variations that are available to soil microbiota. Sorry for my voice. I'm caught with this cough and cold, but I hope I'm audible to you. So,
यस सर
there are bacteria like Pseudomonas, Bacillus, various enteric pathogens. If it is a sewage contaminated soil, and then if the soil is contaminated with some kind of pollutants, whenever we say it may be just plastic, it may be a chemical, nearby chemical factory, suppose a chemical factory making BPA, bisphenol A. So their effluent will contain a lot of residual bisphenol A that may be going through the drain, and the drainage will be contaminating nearby soil. Now, soil received these chemicals, bacteria are growing in those soils, So in order to survive, they will look for some metabolic pathways in order to degrade these pollutants. So there will be a very good chance that you will get bisphenol A degrading bacteria in such contaminated soil sample, right? Because in the terms of carbon sources, bisphenol A is the most available carbon source, right? So the bacteria that will be able to consume or degrade these type of compounds, they will be happily growing and multiplying there in the soil. So their number will be more. So this is the philosophy people take up when they look for oil degrading bacteria. They will look for oil contaminated site, uh, any kind of chemical pollutant degrading bacteria. They will look for those kind of chemical pollutant soil sample, right? And in addition to that, you may have heard of this mycorrhizal communication going on in the forest. That is called mycorrhizal communication or network in the forest, where trees communicate through the hypha, fungal hypha that may be 2 meter to 2 kilometer long. Fungal hypha, they works as a internet or web communication between various trees so that they can exchange information about nutrients and stress signals. So this is quite a unique study that has gained momentum in recent years. All is associated with soil microbiota.
Hello, hello, Dr. Prasun.
Yes, Arvind sir. Yes, sir.
Yes, yes. So I'm very happy. I was listening to you also, and today many participants have joined.
Yes, sir.
So yes, yes, yes. And a very popular topic today we have taken soil
Yes,
microflora. Why I'm telling popular? Because every type of microorganism you will get in the soil. You search for proteolytic, you search for lignolytic, you search for cellulolytic, you search for thermophilic, you search for biofertilizer, means what's so any damn microorganism is always present in soil. That's why if anyone is interested for doing PhD on soil microorganism, there is a tremendous scope. Many scientists, particularly in the agricultural microbiology, they have explored various type of microorganisms. Just Prasun sir was telling actinomycetes. Now they are called as actinobacteria. These actinobacteria produce a geosmin. And in the last week, one student told me, sir, we have taken a research project. We have isolated actinobacteria from the soil. And now we have synthesized the geosmin. And by extracting geosmin, we have prepared scent bottles, which can be sold in the market. That is called as a bio innovation. So ample scope is there for the students. Many students have joined today. So there is ample scope for the research on the soil microbiology topic. I can talk hours and hours together, but I will control myself. And now anyone want to ask the question or give his opinion, his research output. Now topic is again over to Dr. Prasun sir. Please go ahead.
yes. Thank you. Thank you so much, sir, for joining in and enlightening us. Yes, so as sir mentioned that the compound geosins, right, sir, that is in high demand in Arabian market as well. So people make perfume
Hello, sound. You got muted, Prasun sir. You got muted. Huh?
Sorry, sir. Yes, yes. Got to know. So, there are these scent compounds that actinobacteria produces, and that can be sold in the market. In addition to that, sir mentioned very unique point about enzymes. So, several enzymes or enzyme producing bacteria have been isolated using soil microflora. Most of the enzyme industrially active enzymes are produced by fungal isolates. Those are isolated from natural soil sample. There are n number of enzymatic activity that we can look for. It can start with amylases for starch degradation, proteases for protein degradation, lipases, and most importantly, now as we move towards bio E3 economy, so we also look for cellulolytic organisms, those that can hydrolyze or degrade lignocellulosic material, agricultural residues. So agricultural residues are made up of lignin, hemicellulose, and cellulose as the abundant portion. Cellulose degradation will give us sugar, and that sugar can later be used for any other purpose. So people are trying to look for high cellulolytic organism that can release enzymes which degrade these cellulose component of 2G residues. So when we say 2G residues are basically agricultural residues. So now you can imagine that forests have a lot of leaves and their twigs that are degrading naturally. So those will be the sites that may have organisms or microbes that will be able to degrade them. Right. So all cellulolytic organisms can be found in such dam sites. Right. So I hope students may have got this information. Now, in addition to speak one-sidedly, I would like other students to join in. Maneet, are you available?
Yes,
Maneet,
sir.
Nikita Jain. Maneet, yes, yes. Tell me something about your background and your work related to this topic, Maneet.
Namaskar, sir.
Namaste ji.
Sir, I am Manit Sagla, son of Sri Magaramji Sagla from Jodhpur. I enrolled in BTech Biotechnology in first year in MIT Institute of Biotechnology, MIT University, Rajasthan.
Okay, great. So you are a BTech student, right?
Yes, sir.
So you are a BTech first year student, right?
Yes, sir.
So do you have project based learning program in your curriculum?
Yes, sir.
Yes. So to all student participants, I would like to inform that they can explore soil microflora topic
Sir.
because it is very rich in diversity. You believe it or not, you will see when through as you go in your courses, as you spend time in your course, you will realize that various different bacteria. You will learn about taxonomy. Okay, so there are different phylum, different class, different order of organisms, and there are genus and species. Every genus
Sure,
have
sir.
different function. Yes. So try to explore them. Very easy to isolate from an organism and any kind of nutrient media. You can start with nutrient agar media. Use serial dilution method and check what are the microbes that grows in that, right? And then target any one activity, maybe amylase activity or maybe lipase activity, and then you can screen what type of organism is secreting a lot of that enzyme. So suppose you got a very strong amylase producing bacteria, then it can be a good industrial producer. You should work on it and explore more and optimize the process. You may get a product out of it. So, being a BTech biotech
Yes,
student, I think such exploration is very much demanding for your course. At least by the end of your course, fourth year, you should try to characterize one such bacteria.
yes, sir.
Yes, you were saying something. Yeah. Manik, you were saying
Yeah.
something. Yeah. So, you have done this isolation process, microbe isolation process using agar, using nutrient agar. Have you done any kind of isolation in the lab? Manik,
Yes, sir.
have you isolated any bacteria in your lab work, laboratory?
So अभी तक तो लैब
Lab
विजिट नहीं की है, अब करेंगे.
Okay, okay. You are just beginning. Okay, no problem.
Yes, sir.
So, just consider these points, whatever we are discussing. I hope it will be helpful to you.
Sure, sir.
Great. And my number is there. You may anytime get in touch with me in case you want any kind of information from my side. Next, I would like to call upon Yashika Sharma if she is available. Yashika, Garvit Singh Sisodia, Nikita
Yes,
Jain.
sir. Yes, sir.
Okay, Yashika. Yes, Yashika. So tell us
Yes, sir.
something about your background.
Sir, I am first semester student in BTech Biotechnology, MET University, Jaipur.
Okay, so you are batchmate, you and Manik.
Yes, sir.
Great, great. Glad to know young participants, those who are interested to learn more on soil microbiome. I hope you learned two new things I have mentioned besides the conventional things. Okay, so you should always look for the upcoming area like exoelectrogens, like the perfumery sections that people have started working on. Okay, so these are some upcoming field on which new or young minds like yours should work on. So as you learn the new techniques during your course, you should also keep thinking about the industrial aspect. So far, I know both of you are BTech program student. So this means you are going to apply some knowledge that you gain through the course, right? You will have technical degree. So try to find out or come up with a product so that you can also initiate some kind of startup if you will be interested in future. Okay, so this type of discussion will help you broaden your gauge and identify your field of interest. Okay, thank you, Yasika, for joining in.
Yes, sir.
Okay, next, Garvit Singh Sisodia. Garvit, are you available?
Yes, sir.
Yes, Garvit, tell us something about yourself and
Good
your
evening,
work.
sir.
Yes, good evening.
My name is Garvit Singh Sisondia, and my father's name is Mr. Raghvin Singh Sisondia, and I am
Mm-hmm.
belonging from Etah, Uttar Pradesh.
Okay, so you
अभी
are
सर मैं
बोलो.
एमटी यूनिवर्सिटी से राजस्थान से सर मैं बीटेक बायोटेक में एनरोल हूँ सर.
Okay, okay, great.
यस
So
सर.
I think the same thing holds good for you as well. Just like I told Jessica and Manit. So glad that young minds like yours are joining in and trying to learn new things. We are very happy and keep joining such session. It is meant for you guys only because when we will be discussing, you may interrupt us with your questions. How actually we are identifying bacteria. Okay, since you are beginning, you may not be knowing things. So you should ask if you are hesitant, drop the question in the chat box. We'll discuss those points, okay, one by one
यस
as
सर.
we receive them. Okay.
Yes, sir.
So, yes, I am very glad that you people are joining. Thanks, Garvit.
Thank you, sir.
So, Nikita Jain, Satish, Shravani, any one of you available online?
Good evening, sir. Sir, I am Nikita Jain. I am studying MSc Industrial Microbiology from Amity University, Rajasthan, sir.
Very good, very good. So, your specialization is industrial microbiology, right?
Yes, sir.
So, you are first year student?
Sorry, sir. What?
Are you first year student
No, sir.
or
I'm third semester, sir. I'm from third
third
semester,
semester,
second
second
year.
year? Great. Great. Glad to know that you are studying industrial microbiology. So you may have already know all these microbes that are industrially important. Okay. There are various different microbes that are industrially important. The most common one that we know of is Saccharomyces cerevisiae, Zymomonas mobilis, then Lactobacillus, various Bacillus species or their antimicrobial peptides or bifidobacterium for their probiotic nature. So you guys may have heard of Yakult. I will type this in chat box. Yakult is a company. You must go and read about this company and its product. Some of you may be consuming Yakult probiotic drink. So basically, this drink is nothing but a formulation or spore suspension of probiotic bacteria. Okay, so there are various bacteria that may come from fermented foods, that may come from soil as well. Okay, because food we also throw or dump in the form of compost. We do composting in our garden. We may do composting on large scale, like municipality do composting of the wet waste in a designated area. Right, so all those area may have health beneficial microbes, beneficial in the sense of health benefits. Okay, so probiotic organism can be there that can be isolated from such soil microflora. When we say soil microflora, all the microbial diversity present in the soil. And again, compost soil will have different microflora. Sewage sample or mud sample will have different microflora. Arid soil will have different microflora. In our last roundtable meeting, we discussed about uh, bacteria types that grow in least nutrient condition, means very minimal nutrient they will require for their growth. Okay, so there can be various type of organism and their function. And as you are industrial microbiology student, you should always think of organisms that are industrially important or that can be tapped. Their activity can be tapped for the benefit of industry. So when I was talking about enzymes, amylases are very good source, okay, for their activity. So they are being used in detergents, in soap solution that you use in for cloth cleaning. For tannery industry, there are other set of enzymes that people use. So enzymes are utilized in various industrial sectors. Okay, so you should keep looking for such microbes and learn about them. Okay, so thank you for joining in.
Thank you, sir.
Yes, yes, Nikita. Any question you have, any participant, if they have any question or if they have isolated any microbe so far, they may discuss those points here.
सर कैन यू
So,
जस्ट रिपीट कि हम कार्बन सोर्स कौन सा यूज करते हैं ऑयल डिग्रेडिंग बैक्टीरिया में और जो फंगल हाइफे के कम्युनिकेशन रेंज आप रिपीट कर पाओगे?
yes, sure, sure. Thanks, Swapnil, for asking this. So, excuse me. So when we talk about oil degrading bacteria, oil is composed of means take an example of vegetable oil. There can be triglycerides. Triglyceride structure, if you see, there is a glycerol moiety in which on which three different fatty acids are joined. Okay, they are called triglyceride. So in oil, there can be free fatty acids or there can be triglycerides. So these are the carbon sources available for bacteria in that oil contaminated sites because they are contaminated with oils and oil are composed of these compounds. Now, the microbes that will be having lipases or fatty acid degrading enzymes, okay, they are called alkane degrading enzymes may be present or fatty acid degrading enzymes may be present. So, such bacteria can be used to degrade these oils. So now, since the soil itself is contaminated with oil, what we can assume is we will get a unique microbe that will be able to degrade it very fast. And since it will be able to degrade the oil type, this means they will be happily growing in that area. Their number will be more. So isolation of such bacteria will be very easy in such site. So when you are going to isolate such organism, what you will do, you will take the soil sample, you will enrich that type of organism by adding more oil to that soil sample and give the ambient condition, suppose 30 to 37 degrees centigrade, you are working or trying to find out bacteria. You keep the pH between 6.5 to 7.5. Okay, allow them to grow and in case they may get into stress, so they you can supply 0.25 to 0.5% of glucose just to support their growth. Okay, if you want to be very strict that you don't want other bacteria to grow, you don't supply glucose at all. Okay, you just give them oil, enrich them in the lab. Okay, and you subculture it. Subculturing means you take suppose 1 gram of oil contaminated soil, you mix it with 100 ml of buffer or any saline solution, keep it in 37 degrees centigrade for say 1 day. Then you transfer 1/10 part of this culture to another tube or another flask. Again, culture it for 24 hours. Then again do the subculture. By doing this, you are trying to enrich only the bacteria that will be able to consume oil as a carbon source. So only those bacteria will survive that will have ability to degrade the oil, right? So by this way, you can isolate oil degrading microbes. So after 3 to 4 cycle, you can plate them on a nutrient agar plate with supplemented with the oil that you want to degrade. Okay. And then see the growth of the organism. The colony that is growing very fast is the most possibly the most suitable organism for your work because it is able to grow in that environment because you have not given any other carbon sources and it is growing in the oil itself. So it must be able to consume the oil and grow. Right. So I hope I am clear how you can isolate an oil degrading microbe. Then later on you can check with the enzymes they are secreting, how they are degrading the oil and what is their activity, means how fast they are able to degrade the oil. Recently I isolated surfactant producing Pseudomonas strain from petroleum polluted means petroleum soil, petroleum contaminated soil from a nearby petrol pump where they are regularly fueling, doing the fueling. So oil gets dripped in the nearby soil. We took those soil and isolated Pseudomonas originus strain and fed this culture later on. But what we found out that this culture was able to produce very high quantity of surfactant. Surfactant is nothing but means amphipathic molecule having both the hydrophilic and the hydrophobic part. So they can work as a solubilizing agent, oil solubilizing agent, so that bacteria can take up more and more amount of oil and degrade it. So oil can be uh, turned into micelles, small micelles through these surfactants, and then these micelles can be taken up by bacteria such that the oil can be degraded. So surfactant molecule producing bacteria can also be isolated. Oil degrading bacteria can also be isolated from such sites. Now the next question that you asked is about the fungal mycorrhizal network. So it was a ecological study done by Canadian scientists, and after they first uh, said that uh, two plants growing in a quite distance, means around 100 to 500-meter distance, they were able to communicate using fungal hypha. Fungal hypha, if you know, fungus can grow in filamentous form as well as like unicellular form as well. So, in nature, there are various mycorrhizal fungi that grow in long hypha. So, within these hypha, there will be nuclei, polynucleated hypha will be there, and exchange of nutrients happen through this hypha. Hypha size can be of 1 millimeter, can be of 1 meter, and through this study, they find out that several different fungal hypha they get interconnected and they can reach to a distance as long as 100 to 200 meter, and some scientists claim that it can reach to 2 kilometer even. So there are debates on the length or the reach of these hypha because fungal hypha are very minute. Their diameter is maybe just 3 micron or less than that. So they are very minute structure. So studying them is very hard. But people uses fluorescent molecule or molecule that they can later on detect within the soil sample directly. So they use such microbes, such fungal or mycorrhiza, uh, on which they can study the communication. So what they find out that some amount of nutrient is passing from one tree to the other. So the other tree was basically nutrient deprived, and this tree which was having a lot of nutrient was supplying the excess nutrient through this fungal hyphae to the other plant. And during this process, fungus is also taking some amount of nutrient. So it is a kind of symbiotic relationship between plant and fungus. And this is how in the forest, different plant species they communicate with each other, and it is speculated that through this only in the forest a particular plant species survive for a longer period of time. When I am saying longer period of time, this means you are talking about 100 years or more than that. Okay, so one plant species can communicate to the other, and by this way they can also sense the availability of nutrients in any particular area. They can save other species as well. Uh, Shravani, thank you for letting me know, but my mic is unmuted and I hope I am audible. Other participants may also mention if my voice is not reaching to you. Swapnil, am I audible?
Yes, sir.
Okay. Uh, Thank you, Twinkle. Thank you, Swapnil. So, Shravani, I think you check your speaker because from my side, my mic is unmuted.
सर वन मोर
Okay.
क्वेश्चन कि
Yes.
ये जो हम लोग बैक्टीरिया जो है वो ऑयल डिग्रेड कर पाते हैं तो टेक्निकली उसको तो हमें जो ऑयल स्पिल हो जाता है पानी में उसमें यूज़ करते हैं, तो क्या हम करते हैं या ये लार्ज स्केल पर नहीं हुआ है कि ऑन वर्किंग प्रोसेस है?
बिल्कुल, very good question. Very nice. Even in the last meeting also, your question was very good, and uh, other participants are also encouraged to ask such thing. So to answer you, when there is some kind of oil spillage, recently two oil spillages happened. So far I remember, one happened in the Bay of Bengal. Okay, recent oil spillage happened. So oil spillage, when we are saying India buy a lot of oil. You may have been listening to the news that we are buying oil from Russia. We are also buying oil from Middle East countries, okay, OPEC nations. So this means these oil are coming in tankers through ships, and some ships they if they crash in the sea, so oil gets spilled in the ocean itself. Oil being less dense than water, they float on the top. And you know that sea is not silent. There are waves, so oil spillage, it floats across the sea. Okay, it may go around here and there, and it hampers the aquatic diversity. So what people try? People try to uh, remove this oil spillage. So your question is, can we use such surfactant molecule or such bacteria to degrade oil? Now there are two organisms that people have identified. So first, I will address this question through microbial perspective. Then I will say how naturally people are doing on large scale, because using microbe in a sea is a kind of futile exercise, but that can be done. So there are two microbes you may have heard or you will read in your textbook about Pseudomonas putida. Pseudomonas putida can degrade various kind of phenolic as well as non-phenolic compound, including oil. Okay, so Pseudomonas putida strain. Uh, there is one scientist, Indian scientist, who identified and genetically modified this strain and explored it a lot using plasmid-based genetic engineering. So they incorporated a lot of oil-degrading metabolic routes through plasmids, and they developed Pseudomonas putida strain, which can degrade various different types of oil. Then there is another scientist who just now recently retired. His name is Dr. Banwari Lal. He identified a unique bacteria. He termed that because it is a patented bacteria. Later on, they patented these two organisms that they made, and they are producing this in large quantities in the name of oil zapper. I'll write this down. Oil zapper. Okay, so they are making immobilized culture of these oil zappers, two different bacterial isolates, and they are selling this to Middle East countries. They are selling this into Middle East countries. Now imagine when they are selling it to Middle East countries, it is not just 1 kg or 2 kg of bacteria. They are produced in 5,000 liter capacity tank. Only they will culture the bacteria, then they will dry them, make the formulation in dry form, and immobilize them on some kind of support or matrix, and it will go to oil manufacturing or oil isolating countries. So they are having oil plants. So wherever they are pumping oil, these Middle East countries, they are pumping out oil from the soil. So they will have a lot of soil area or a lot of land which are contaminated by these oils, right? And they want these oils to be removed, okay, because slowly and slowly it will make the land infertile. So these kind of oil jappers are used, and Middle East nations are buying these oil jappers in tons of quantity every year. So these are the aspect of industrial microbiology from soil microflora point of view. Now coming back to the question that whether it is literally feasible in a oil spillage site that has happened in sea. So in the sea, due to the waves, it is quite impossible. So what people do, people basically float plastic bound or floating materials as the oils. So we can use sponge kind of thing that can float around. We can make a circle. Circle means very huge circle that is uh, as large as 3 to 5 kilometers long. Okay, and then try to drag the soil and make a small pond within the sea itself, and then collect the oil from the top using a pump. While I am saying things like that, also remember that these oils are flammable. Okay, so they may catch fire in an open space. So we have to consider that point as well. So currently the easiest practice was means either it by nature they take up the fire, or if fire has not happened, we try to pull it out using pumps and using such kind of spongy material. Okay, people are trying to develop immobilized bacterial system. So bacteria will be immobilized in these sponges itself, and they will slowly degrade the oil. Mind also keep in mind that when we are seeing microbial process, these process are a bit slow. It is not that bacteria will eat up 1 liter of oil in a day. Okay, so a lot of bacteria will be required to degrade 1 liter of such oil. So the process of degradation is quite slow. If we can modify enzymes that can increase this processing time to suppose 10 hour or 5 hour, then such thing can become very interesting, and then such thing can be applied on a large scale in a real field application. I hope I am clear to your question, Swapnil.
Yes, sir.
Yes. So I think it is too much time. I would like to wind it up, but before winding it up, to all the students and the participants, if they have any question related to soil microflora, they can mention here or in the chat box.
Good evening, sir. Sir, I have a question.
Yes, Tim.
Sir, can you tell me about which database is used to study soil microflora?
Uh, I have not gone through such databases, but I am sure that people are making secondary databases. Secondary databases comes from primary database. Okay, so primary
Okay.
database are like right. Sorry, I hope I am audible. So primary databases like NCBI can also be explored. So what I mean to say is NCBI has data for all the microbes isolated till date, characterized till date. Okay, then secondary database get data from NCBI only. So you can filter out using keywords like, so you can give the keyword using the advanced search option in the NCBI. You use the term oil, use the term and use the term degradation. So oil and degradation. Then if you know about the genus of the bacteria, mention the genus. If you don't know about any such thing, just use oil and degradation and let it search. You will see the output. Then you can screen what are the output, what are the organisms coming in the output, and you can check whether they are well known or not using Google Scholar or using the data that is given in the database itself. Okay. So far, to my knowledge, Pseudomonas species is the best one, whether it is Pseudomonas aeruginosa or any other species of Pseudomonas. Okay, they are very versatile organisms. I hope
Okay, sir.
I
Thank you
addressed
very
your
much.
question. Yes. Thank you, Twinkle, for joining in. And to all the participants, I have dropped a message in the chat box. You can see the chat box. There is a link available. So it is a kind of feedback form as well as a certificate that you have participated in this round table discussion. Okay, so you all are requested to fill this form and what are your expectations? What you want means any topic you want to be covered in such platform. Okay. Also, we would like to inform you that okay, 17th of September is World Microorganism Day. 17th of September. So do celebrate this day. Being a microbiology student, do celebrate this day if you have student unit running in your department. So with this, I thank you everyone to join this session. I hope this session was fruitful and do send us your feedback because we will improve, we will help you grow and get the knowledge and your participation, your questions, your interaction will will help you as well. Okay. So with this, I say goodbye to you. Okay. Thank you so much. Anybody having problem in finding the link, they can mention it now itself. Otherwise, I will end this meeting. Thank you, Twinkle, for your appreciation. Thank you, Nikita Jain.
Thank you so much, sir.
I hope you are able to. Yes, welcome, Bachi. So I hope you all can see the Google Form link because if I end this link, you may not see the chat. So better you can click that link or save it for later link. Okay. So I'm keeping this link, this meeting live for 5 more minutes. After that, I will end myself. Okay. Thank you. Thank you, everyone.